It's a simple question. Can we edit our genes with CRISPR safely? The answer is not so simple. It's not a yes or a no. It's more like a, it depends. It depends on what we're editing. It depends on who we're editing, and it depends on what we mean by safe. Let's start at the beginning. What is CRISPR? You've probably heard it described as molecular scissors. That's a good way to think about it. It's a tool that can find a specific spot in our DNA and make a cut. Our DNA is like a massive instruction book. It has billions of letters. These letters tell our bodies how to build and run everything. Sometimes there's a typo in that book. A single wrong letter can cause a devastating disease like sickle cell anemia or cystic fibrosis. For a long time, we could read the book, we could find the typos, but we couldn't fix them. It was like knowing exactly what was wrong with a car's engine, but having your hands tied behind your back. You could diagnose, but you couldn't repair. Then came CRISPR. CRISPR is actually a natural system. It comes from bacteria. Bacteria use it as an immune system to fight off viruses. They store little snippets of viral DNA, like mugshots. If that virus attacks again, the bacteria sends out a protein, often one called Cas9, carrying that mugshot. The protein scans all the DNA it finds. When it finds a perfect match, it cuts the viral DNA, destroying the invader. Scientists realized they could hijack this system. They could give the Cas9 protein a custom-made mugshot, one that matches a human gene. They could tell it, go find this specific sequence in human DNA and cut it. This was a massive breakthrough. It gave us the power to edit the instruction book. When CRISPR cuts the DNA, the cell's natural repair crew rushes in to fix the break. The cell has two main ways of doing this. One is quick and dirty. It just glues the two broken ends back together. This process is often messy and can lead to small errors, like adding or deleting a few DNA letters. This is actually useful if you want to turn a gene off. By messing up its instructions, you can disable it. This is called a gene knockout. The other repair method is more precise. If there's a template nearby, a correct version of the DNA sequence, the cell can use that template to fix the break perfectly. The scientists can supply this template. They can give the cell a piece of healthy DNA along with the CRISPR machinery. When the cut is made, the cell's repair system sees the template and uses it to rewrite the faulty gene. This is how you could, in theory, correct a typo that causes a genetic disease. So, we have a tool that can find and replace faulty genes. The potential is stunning. We're talking about fixing diseases that have plagued families for generations. Diseases that, until now, were a life sentence. Let's talk about where this is already happening. Let's talk about Victoria Gray. She had sickle cell disease. It's a brutal condition caused by a single typo in the gene that codes for hemoglobin, the protein in red blood cells that carries oxygen. Her red blood cells, instead of being round and flexible, would become hard and C-shaped like a sickle. They would get stuck in her blood vessels, causing excruciating pain, organ damage, and strokes. Her life was filled with hospital visits and blood transfusions. The disease was a constant shadow. In 2019, she became one of the first people to be treated with a CRISPR-based therapy. The doctors didn't edit every cell in her body. That's still incredibly difficult. Instead, they took some of her stem cells out of her bone marrow. These are the cells that create new blood cells. They took them to a lab. In that lab, they used CRISPR, but they didn't try to fix the sickle cell typo directly. They did something clever. They used CRISPR to turn off a different gene, a gene that acts like a switch. This switch, called BCL11A, normally turns off the production of fetal hemoglobin right after we're born. Fetal hemoglobin is a version we all make in the womb, and it works perfectly. It doesn't cause sickling. The plan was to use CRISPR to break that switch. By turning off the BCL11A gene in her stem cells, they could trick her body into producing the healthy fetal hemoglobin again. They edited her cells outside her body, in a dish. This is called ex vivo editing. It's a major safety feature. It allows them to check the cells, make sure the edit worked, and screen for major errors before putting them back. After editing the cells, they gave Victoria a high dose of chemotherapy. This was to wipe out the old, faulty stem cells in her bone marrow to make room for the new edited ones. Then, they infused her own edited cells back into her body. The wait began. Would the edited cells take root? Would they start producing new healthy blood cells? They did. Her body started making fetal hemoglobin. Her sickled cells were replaced by healthy, round ones. The results were life-changing. Her pain crises stopped. She no longer needed blood transfusions. She could live a normal life. She could play with her kids without fear. CRISPR didn't just treat her symptoms. It went to the source of the problem, the DNA itself, and fixed it. This is a powerful argument for, yes, it can be safe. For someone like Victoria Gray, the risks of her disease were enormous. The potential benefits of the therapy were also enormous. The treatment itself had risks, like the chemotherapy, but the CRISPR part, done ex vivo in a controlled lab, worked. And it worked for others with sickle cell and a related blood disorder, beta thalassemia. The success of these trials led to the first ever approval of a CRISPR-based therapy in the UK and the US in late 2023. It's now a real medicine for a specific group of people. Similar stories are unfolding for other diseases. There are clinical trials using CRISPR to treat a form of genetic blindness. In that case, the CRISPR machinery is injected directly into the eye to try and correct the faulty gene in the retina cells. This is in vivo editing inside the body, which is a bit riskier because you have less control. But the eye is a relatively contained environment, which limits where the editing tool can go. Early results have been promising, with some patients reporting improved vision. There are also trials for certain types of cancer. The idea is to take a patient's immune cells, called T cells, out of their body. Then use CRISPR to engineer them to be better cancer fighters. They can edit the T cells to make them target cancer cells more effectively and to resist being shut down by the tumor. These supercharged cells are then infused back into the patient. In these specific, highly controlled medical situations, CRISPR is looking safer and safer. The process is overseen by doctors and regulators. The patients are people with severe diseases who have few or no other options. The risk is calculated. For them, the known danger of their disease is greater than the potential danger of the new treatment. But this brings us to the other side of the coin, the "it depends" part. What are the dangers? Why isn't CRISPR being used to fix every genetic disease right now? The biggest and most talked about risk is something called off-target effects. The molecular scissors are good, but they're not perfect. Our DNA is vast, with 3 billion pairs of letters. Sometimes, a sequence in one part of the genome looks very similar to the target sequence. The CRISPR system can get confused and make a cut in the wrong place. What happens if it cuts in the wrong place? It could be harmless. Many parts of our DNA don't seem to have a critical function. But it could also be catastrophic. What if it cuts in the middle of a vital gene, a gene that is supposed to prevent tumors from growing? You could inadvertently disable a safety mechanism your body needs. You could, in theory, cause cancer while trying to cure something else. This is not a hypothetical risk. Scientists have seen off-target cuts happen in lab experiments. Researchers are working tirelessly to make CRISPR more accurate. They've developed new versions of the Cas9 protein that are less likely to make mistakes. They've designed better guide RNAs, the mugshots, that are more specific to their target. They use powerful computer programs to predict likely off-target sites and then test for cuts in those places. In the sickle cell therapy, they chose a target sequence that was known to have a very low risk of off-target effects. So the risk can be managed and reduced, but it's very hard to say it's zero. There's another risk that's less talked about, but just as important on target effects. This means the CRISPR system cuts in the right place, but the cell's repair job is a mess. Instead of a small, neat edit, the cell's repair machinery can sometimes cause large deletions of DNA around the cut site. Or it can cause complex rearrangements, where bits of the chromosome get shuffled around. These large-scale changes can be just as dangerous as an off-target cut. They could delete a neighboring gene or disrupt how a gene is regulated. It's another source of unintended consequences that scientists have to watch for. And then there's the delivery problem. For ex vivo therapies like the sickle cell treatment, it's not an issue. You have the cells in a dish. But for many diseases, you need to edit the cells inside the body. Think about muscular dystrophy, which affects muscle cells throughout the body, or Huntington's disease, which affects brain cells. You can't take all those cells out, you have to get the CRISPR machinery to them. This is a huge challenge. How do you deliver a package containing a protein and a piece of RNA to a specific type of cell, say a liver cell, while ignoring all the kidney cells and heart cells? The most common delivery trucks are viruses. Scientists can take a virus, strip out its own harmful genes, and load it up with the CRISPR components. They use viruses because they are naturally good at getting into our cells. The adeno-associated virus AAV is a popular choice. But using a virus as a delivery truck has its own risks. Our immune system is designed to attack viruses. It might attack the delivery truck before it even reaches its destination, making the therapy ineffective. Or it could cause a massive immune reaction, making the patient very sick. In a few gene therapy trials not using CRISPR, but using similar viral delivery methods, this has had tragic consequences. There's also a small risk that the virus could insert its genetic cargo into the wrong place in our DNA, again with the potential to disrupt an important gene. So, for somatic cell editing editing the cells of a person's body to treat a disease they already have, the answer to the safety question is nuanced. For a life-threatening disease with no other cure, and when done in a controlled way like ex vivo editing, the benefits can outweigh the risks. It is becoming a safe enough option, but for less severe conditions or for in vivo editing, the safety bar is much higher, and we're not there yet. We're still learning about the long-term effects. What happens 10, 20, or 50 years after someone has millions of their cells edited? We don't know because the technology is too new. Now we have to talk about the other kind of editing. The kind that changes everything. Germline editing, somatic editing, everything we've talked about so far affects only the person being treated. The changes are not passed on to their children. If Victoria Gray has more children, they could still inherit the sickle cell gene. Germline editing is different. This means editing the DNA in cells that can be inherited sperm, eggs, or an embryo itself. If you make a change here, you're not just editing one person, you're editing their children and their children's children and every generation that follows. You are changing the human gene pool. This is where the conversation about safety becomes intensely difficult and deeply ethical. The potential benefits seem extraordinary. You could, in theory, eliminate a hereditary disease like Huntington's from a family line forever. A single edit could spare countless future generations from suffering, but the risks are astronomical. All the dangers we discussed, every off-target effects on target errors, are magnified infinitely. An off-target cut in a somatic cell might cause a tumor in one person. An off-target cut in an embryo would be present in every single cell of that person's body for their entire life. And it would be passed on to their children. A mistake wouldn't just harm one person, it would create a new heritable disease. We simply do not know enough about the human genome to predict all the long-term consequences of such a change. And gens often have more than one job. A gene that contributes to one disease might also play a subtle, crucial role in brain development or immune function fixing. One problem could create 5 others that we won't discover for decades. And we have a terrible, real-world example of what happens when someone rushes ahead. In 2018, a scientist in China named He Jiankui announced that he had created the world's first gene-edited babies. He had edited the embryos of twin girls. His goal was to make them resistant to HIV. He targeted a gene called CCR5. Some people have a natural mutation in this gene that makes them highly resistant to the virus. He used CRISPR to try and recreate this mutation in the embryos. The scientific community reacted with horror. His work was unethical, irresponsible, and medically unnecessary. There are far safer ways to prevent HIV transmission. His science was also sloppy. The editing didn't work perfectly. The girls didn't have the precise known mutation that confers HIV resistance. Instead, they had new, never-before-seen mutations in the CCR5 gene. We have no idea what these new mutations will do to their health over their lifetimes. Furthermore, the editing was mosaic. This means that not all the cells in their bodies had the edit. Some cells were edited, some were not, and some were edited differently. It was a chaotic, unpredictable mess. He, Jiankui, broke every rule of scientific and ethical conduct. He acted in secret, without proper oversight, and on a problem that didn't require such a drastic solution. His experiment didn't make the babies safer, it exposed them to unknown and unknowable risks for their entire lives, and for the lives of their potential descendants. He was eventually sentenced to prison, but the damage was done. The 2 girls, and possibly a 3rd child, are alive. They are the first humans with an edited germline. The He Jiankui affair was a brutal wake-up call. It showed what can happen when this powerful tool is used without caution, without humility, and without a deep sense of ethical responsibility. As a result, there is a broad international consensus among scientists and ethicists. We should not perform clinical germline editing at this time. There is a moratorium, a temporary ban. The technology is not ready, the risks are too high, and we as a society have not had the necessary conversations about whether we should ever do it, even if it becomes perfectly safe. Because what happens if we can do it safely? What happens if we develop a version of CRISPR that is 100% accurate with no off-target effects? The ethical questions don't go away, they get harder. If we can eliminate Huntington's, should we? Most people would probably say yes. What about the genes that predispose someone to Alzheimer's or breast cancer? Probably yes again. But then the line starts to blur. What about editing genes related to height? Or muscle mass or intelligence? What about eye color? This is the designer baby scenario. The fear is that gene editing could become a tool for enhancement, not just for therapy. It could create a world of genetic haves and have-nots, a world where the wealthy can afford to give their children genetic advantages, creating a biological caste system. This is not science fiction, it's a real social danger that we would have to confront if germline editing ever became routine. Who decides what is a disease to be cured and what is a natural human variation to be accepted? Is deafness a disability to be fixed, or is it a cultural identity for many in the deaf community? These are not scientific questions. They are human questions, and we don't have the answers. So where does that leave us? The path forward is being forged in two very different ways. For somatic editing number treating existing diseases in individuals, the path is one of careful incremental progress. Scientists are developing better, safer versions of CRISPR. There's something called base editing, which doesn't cut the DNA double helix. Instead, it acts more like a pencil and eraser, directly changing one DNA letter into another. There's also prime editing, which is even more precise, like a search and replace function in a word processor. These newer technologies promise to reduce the risk of off-target and on-target errors significantly. At the same time, regulatory bodies like the FDA in the United States are creating rigorous frameworks for clinical trials. Every new therapy has to go through years of testing, first in lab animals and then in small, carefully monitored groups of human patients. The approval of the sickle cell therapy was a landmark, but it was the result of a decade of research and a cautious, step-by-step process. This is how we make medicine safer. It's slow, it's expensive, but it's necessary. For germline editing, the path is one of global conversation and restraint. Scientific bodies are calling for public debate. They are trying to establish international norms and regulations to prevent another rogue experiment like Hijankui's. The consensus is that we should continue basic research in the lab on isolated cells and animals to understand the technology better. But we should not cross the line into creating a gene-edited human being until and unless there is a broad societal agreement that it is safe and ethically acceptable to do so. And that agreement may never come. So, can we edit our genes with CRISPR safely? For a person with a devastating genetic disease with no other hope, the answer is a qualified yes. We can now, under strict medical supervision, take cells out of their body, edit them to fix the problem, and return them. The risks are real, but they are becoming manageable and are often outweighed by the certainty of the disease. We are at the very beginning of this medical revolution, but it has begun. For the human species as a whole, for the idea of editing our children and all future generations, the answer is a firm no. Not now. It is not safe. We don't know enough. The tool is too powerful, and we do not yet have the wisdom to wield it. The biggest danger of CRISPR isn't that it might make a mistake and cut the wrong gene. The biggest danger is that we might make a mistake and cut ourselves off from our own humanity. Safety isn't just about technical precision, it's about ethical clarity, and on that front, we still have a long way to go.